Hello everyone. Today we are going to study the topic colorimetry and spectrophotometry from unit 1st of paper 8, and this is part first of this topic. This is one of the important topics for practicals also, and in this topic we are going to study the introduction, theory of colorimetry and spectrophotometry, principle that is Beer-Lambert law, instrumentation and application of colorimetry and spectrophotometry. So we'll begin with this topic. So colorimetry, as the name tells, it deals with color measurement. It is a type of chemical analysis which uses a selective light absorption. You know that in colorimeter experiment we are using colored solution, and that colored solution will be absorbing light. And in this experiment we will use selective light absorption by different colored solutions as the principle. So measurement of amount of light absorbed by the color developed in a sample. This is the method which is used in the colorimetry. So each sample which will be having different color, that is absorbing light to different extent, and that is measured by using colorimetry. So human eye, it is the best colorimeter in the visible region, and we know that we can see different colors, for example rainbow. So this human eye is said to be a natural colorimeter. The principle involved in this colorimetry is it is the determination of relative absorption of light with respect to a known concentration of a substance. That means if you consider the colorimeter experiment which is given in the practicals, that is colorimetric estimation of copper ions in the given solution, there you can see that we are using different copper sulfate solution. That is, first we will add 2, then 4 ml, then 6, 8, like that. So we will vary the volume of the copper sulfate solution so that we will get solutions with different color intensity. And actually our aim there is to find out the concentration of copper ions which are present in the unknown solution. So for that, in the beginning we are preparing different solution of different concentration so that we will get one graph for that solutions, and from that calibrated solutions we are finding the concentration of the unknown sample which is given to you. So the principle of colorimeter is, or colorimetry is, determination of relative absorption of light with respect to a known concentration of a substance. So what happens when light is incident on an object, especially a homogeneous medium? So when light is incident, there are different possibilities. The first possibility may be reflection, that is, light gets reflected back at the surface itself, that is reflection. Then absorption. So if it is colored solution, whatever the quantity of light that is incident on that medium, that will be absorbed by that medium due to the particles which are present in that medium, and that medium may be a glass medium or it may be a solution also. And there is another possibility, that is, whatever the remaining light which is not absorbed by that medium, that will be sent out, and that is called as transmission, and that light is called as transmitted light. So there are three different possibilities when light is incident on a material object, especially transparent medium, that is reflection, absorption and transmission. So in studying colorimetry we are using the second process, that is absorption. So you know that in practicals also we find the optical density, that is nothing but the absorbance of the medium, or in that case absorbance of the solution. And we can also find the transmittance in case of colorimetry. Either one of them you can find, so that you will find the other one, because absorbance and transmittance, or absorption and transmission, are opposite to each other, or they are inversely proportional. If absorption is 0, if for a solution absorption is 0, then its transmittance will be 100%. If transmittance is 0, then it is understood that no light will be allowed out of the medium. So absorbance will be 100%. So they are inversely proportional. So we'll see the theory of colorimetry or spectrophotometry. So according to that, as we see in the previous slide, the incident light will be sum of absorbed light plus reflected light plus transmitted light, or incident light is the sum of incident light is the sum of absorbed light, reflected light and transmitted light. But if you consider air glass interface, in that case we can neglect that reflection, so that we can neglect the incident reflected light intensity. Okay, so the equation becomes I naught is equal to IA plus IT. Now what is color? So how do you get color, or how do we see color? So whenever there is interaction between light and matter, there is a possibility of seeing color. So mainly to see color, we need light and matter. So you can understand by a simple example. If there is complete darkness, can you see any color except black? No. So that is due to absence of light. Though there is any matter or material object in the darkness, we cannot see the color because there is the absence of light. So to see color, we need light as well as matter. So color results when radiation is absorbed by an element or by a compound formed through a reaction. So if you want to develop a color, especially in case of colorimetric estimation of copper, you can understand there we are putting copper sulfate solution in different volumes, 2, 4, 6, 8 like that. Then you are adding 1:1 ammonia in the same quantity to each different standard flask. So you'll get a color. That color is due to formation of cuprammonium sulfate complex. So color results when radiation is absorbed by an element or by a compound formed through a reaction. So that color of the solution is due to the formation of complex particles, and that particles, or it may be complex ions, which are present in that colored solution, they will absorb the light whenever light is passed into that colored solution, so that we feel that it is colored. So though we see that copper sulfate solution or cuprammonium sulfate solution is dark blue in color, actually it is not the color of cuprammonium sulfate solution, because whatever the color we see, it is the contrary color of the color absorbed by the particles in the solution. That means if a solution looks blue in color, it may be absorbed some other color, okay, before getting blue color. So we'll understand that later. So if you consider one colored solution, don't bother about the color here, just see the colored solution. Think that we are passing a white light which is consisting of red, yellow and blue color, okay, or it contains light radiations of wavelength corresponding to red, yellow and blue. So whenever this white light is passed into a colored solution, think that that solution absorbs red color. So red color will be only absorbed by that colored solution. Whatever the light which is coming out from that, then it will be containing only yellow and blue color. So this colored solution, though it absorbs red color, it won't be seen as red in color, it will be seen as green in color, because whatever the transmitted light, it will be containing yellow and blue color, and that if you mix together yellow and blue, you'll get the green color. So for us, this solution, this colored solution will be visible as green colored solution and not red colored solution, because though it absorbs red color, the output or transmitted light has absence of that red color. So the outsider feels that it is the mixture of yellow and blue color, so it will be visible as green in color. So the quantity of radiation absorbed by the molecule depends on these factors. One is absorbing medium, and frequency of radiation. So whatever the quantity of radiation that is absorbed by the medium or any solution, that depends on that medium or particles which is present in that absorbing medium, or it may be solution also. And also it depends on the frequency of the radiation passed, frequency or wavelength of the radiation passed. So we set that wavelength at the beginning of the colorimeter experiment. You can remember we set 600 nanometer for some experiment. For some experiment we will set 540 nanometer at the beginning in the colorimeter. That is because this absorption is also dependent on the wavelength or frequency of the radiation. So to study colored substance we need wavelength from visible region, and to study colorless substance we need wavelength from UV region. Or you can differentiate like this: to study colored substance we are using colorimetry, and to study colorless substance we can use spectrophotometry. So these are colorimetric instruments which were used in earlier days. Now it is developed, and we have a term called photometer. It is the instrument which is used to measure the intensity of light, and this photometer itself is a part in spectrophotometer. As the name tells, it has spectrophotometer. Spectrometer plus photometer gives a spectrophotometer. So what are the properties of colored solution that must be used in colorimeter experiment? So the first property is stable, then reproducible, then sensitive. So what is meant by stable? That means if you prepare a colored solution for colorimeter experiment, that formed color must be stable. For example, if you prepare copper sulfate solution by adding 2468 ml of copper sulfate and then by adding ammonia, 1:1 ammonia, then you will get a colored solution, dark colored solution, and that formed color must be stable for long time. It should not disappear after 5 or 6 minutes like that. So it must be as it is. So it is called as stable or stability. Next, reproducible. Reproducible means, for example, if you prepare a solution in standard flask by adding 2 ml of copper sulfate and then ammonia today, and if you prepare the same solution tomorrow, then whatever the color intensity, that must be same, because you have used same volume of copper sulfate and 1:1 ammonia solution. So that should not change. If you prepare on the third day also, you should get the same intensity. That is called as reproducibility. Okay. Or you and your friend, if both of them prepare the same solution in the lab, both of you have to get the same intensity. It is called as reproducibility. And sensitivity or sensitive. Whatever the reagents which are used to prepare that colored solution, they must be very sensitive to reaction and they must form the complex which will give the color. That is called as sensitivity. And this color development, it is dependent on pH, time, temperature and ionic strength. So it is dependent on the experiment. So for some colorimetric determination, that solutions are pH dependent. We may be adding acids or bases during the formation of the complex, and some solution may take long time to get colored, and some solution may require temperature to get the color, and also it is dependent on the ionic strength, that is the concentration of the solution which is prepared. So we'll see the principle of colorimeter. When a monochromatic light passes through a colored solution, some specific wavelength of light are observed which is related to color intensity. The amount of light absorbed or transmitted by a colored solution is in accordance with two laws, that is Beat's law and Lambert's law. So if you consider a solution which is taken in a glass medium like this, now if you pass light ray of intensity I0, okay, I0 suggests that it is the intensity of incident light. When such light is passed through this colored solution which is having concentration C, and the path length for this light that is nothing but the distance between this edge to this edge of this beaker or medium. Okay, this is called as path length. It is represented as B. And whatever the light which is coming out after absorption, that is represented as, that intensity is represented as I. So I naught is intensity of incident light, I is the intensity of the transmitted light. And during the course of entering of this ray into this solution, there will be absorption of some of the incident light, because this is colored solution, and you know that colored solution will be containing particles, and that particles will absorb the light, and whatever the unabsorbed light is there, that will be coming out as transmitted light. So the amount of light absorbed or it may be transmitted, that is studied by using two laws. One is Beer's law, another one is Lambert's law, and finally we will combine that to study it as Beer-Lambert